Yeah, so similar to last week's speaker, the last time I was in this room, I was actually defending my PhD dissertation. And then I was talking, of course, about mapping in Arabidopsis, and today I'll be talking about mapping in tomato. In full disclosure, I'm not actually a plant breeder, I'm a bioinformatician, but a lot of the projects that I work on have direct applications to useful agricultural traits. So hopefully that counts. And then also, as Joyce mentioned, I have a little bit of an heirloom tomato obsession. So here is my rainbow of tomatoes from my garden 2 years ago. Last summer wasn't so great because it was a bit wet and various disease problems occurred, but 2 years ago it was great. So those are my babies. So I'll be talking about tomato and also closely related species today. So in this phylogeny, you can see what I'm considering the members of the tomato clade. So there are 13 species in the tomato clade. You can see that one of them is actually missing here, but I'll be focusing on these today. And there's also Solanum like a persicoides, which you can see at the bottom of the tree. So that is also another species we'll be talking about today, which is an outgroup. So the nice thing about these is that they can actually be crossed with varying levels of difficulty. So the ones towards the bottom of the tree tend to have self-incompatibility and also unilateral incompatibility, which can make it a bit more difficult to cross those to cultivated tomato. The ones towards the top of the trees, are tend to be self-compatible. These are mainly found in Western South America, so Peru and Ecuador, as you can see the map here. Let's see if I can get this thing to work. Maybe this, there we go. So here, and there's also 2 species that are found in the Galapagos Islands. This is a little bit blurry, Galapagense and Cheesemanii, which are found solely in the Galapagos Islands. They look kind of similar, the different wild species of tomato. You can see some of the diversity here. So Habricites tends to have a lot of trichomes, so it looks kind of fuzzy. You can see in the middle here, there's some seedlings that we were growing in our greenhouse. So at the bottom, you can see Habricites. The reference tomato is Solanum like a persicum Heinz, so that's growing in the middle of this tray here. I don't think this is really working. And then at the top, we have Pimpinellifolium. So Pimpinellifolium is pretty closely related to the cultivated tomato. You can see there's variation in fruit color and size. So the wild species tend to have smaller fruits. And they also tend to ripen to green. So they never actually change to red like your typical garden tomato. There's also the 2 species from the Galapagos Islands, Cheesemanii and Galapagense, that are orange fruited. So that's kind of unique. And you see here a number of fruit that we got from Roger at TGRC. So he sent us from their fruits that they grow there. So that was really nice for one of our experiments. And you can see the diversity in color. Here's some of our wild species growing in the greenhouse. So you can see that they're indeterminate, so they get quite large. They have this sprawling phenotype as opposed to some of your more common cultivated tomatoes, which are determinate and don't get nearly as tall. So as I said, you can cross the wild species with cultivated tomato, and this turns out to be really useful because cultivated tomato has undergone 3 bottlenecks. So there was the original bottleneck that happened during domestication. And then a second bottleneck happened when they moved to, when they were transported to Europe. And then a third bottleneck occurred when they were transported back to the US for breeding there. So it's estimated something like 5% of the diversity. So there's a huge loss of diversity in the cultivated tomato. And so what's nice is you can take the wild species, which have a lot more diversity and interesting traits, and then you can cross these to the cultivated tomato. And you might think, so here's some more of my tomatoes. You might think that they're, looking at that fruit shape, that there's actually quite a diversity of phenotypes there, but it's actually controlled by a few loci. So few genes lead to sort of this diversity of phenotypes that you see here. So the wild species have been used in breeding for a number of things. So this has been happening for the last several decades. So they can be used in breeding for disease resistance. So you can see one of our collaborators on a project is interested in tomato yellow leaf curl virus. So he has a cultivar that is resistant to that. So you can see in the bottom picture, this line is called GH13. The plants in the back are nice and healthy. They're resistant, and the ones in the foreground are not resistant to tomato yellow leaf curl. And today I'll also be talking about bacterial speck. So that's another trait that wild species have been used to increase resistance to that problem. The wild species can also be used in fruit quality and nutrition, and then some other things like leaf morphology and flowering time. So today I'll be talking about some different projects that we've had, and the common theme in all of these is using bioinformatics approaches to map genes of interest in tomatoes and their wild relatives. So the first thing I'll talk about is defining introgression boundaries. And this started originally as something else. So when I first came to Lucas's lab as a postdoc, we were handed the sequence from Galepagense, Solanum galepagense. So this was an interesting species because, like I said, it came from the Galapagos Islands. So it evolved in relative isolation until recently, except for a couple other species that were found in the island. It's very closely related to like a persicum, and most of the work done at that time, and even now, it's a bit unclear where it falls on the tree. So I'm not really sure why my picture is so blurry, but the Galepagense actually falls here with Cheesemanii, pimpinellifolium, and like a persicum. So it's a little unclear how these species are related whenever you do phylogenies. So we wanted to do some work to try to clarify that using the genome sequence. And also, since we knew that Heinz, which is our reference genome, has pimpinellifolium in its parentage, we also wanted to figure out where in the reference genome, Heinz, we could find introgressions from pimpinellifolium. So in order to do that, we selected an old heirloom line, yellow pear. So you can see another one of my garden pictures there. So this is an old heirloom line that existed prior to modern breeding programs, so it should be free of introgressions. So this was our control, basically. To do this, we devised a pipeline that's adapted from White et al., a paper that came out in 2009. So we had 5 different species that we looked at. So we had the reference Heinz genome that we used to map all of our resequencing data onto. We had the Galepagense pimpinellifolium data that came from the Tomato Genome Project, so that was publicly available. We had our yellow pear resequencing data, and then of course potato sequence was available at the time. So we mapped all these to Heinz, the resequencing data to Heinz, and then we created genomes from these. So we substituted in the SNPs, and any place we had a gap, we masked those out. We also then masked with repeat masker, so we could get rid of any repetitive regions that we couldn't be really sure where they mapped to Heinz. Then we did whole genome alignments with these 5 species. We ran PALP to try to delimit where the phylogenetic breakpoints were occurring along the alignment. Then each one of these partitions, we ran Mr. Bayes on to determine a tree for that partition. And then all these trees were mapped back onto the Heinz genome, so that we were able to see where a change in topology occurred along the chromosome. In conjunction with that, we also plotted the density of SNPs across the Heinz genome. So this was resequencing data. So we used a standard SNP calling pipeline, mapped the data called SNPs. And then we put these into bins of 10 kilobases. And then we plotted the density of SNPs along the chromosome. So here you can see at the top of the diagram or the picture, we have yellow pear. And then the second species is Galepagense, and the third is pimpinellifolium. So if you look at yellow pear, where that red circle oval is, you see there's sort of this little peak in the SNP density. And then if you come down to the last plot, you see that there's a reduction in SNP density there. So that's a putative introgression. And whenever we took that location and then also used our data from the whole genome plots, we found that there was a change in topology. So you would expect Hines and yellow pear to be clustered together in the tree since they're actually both from Lycopersicum. So that's sort of the expected topology. If you move on to topology 2, you see that it's flipped. So now Hines is actually closer to pimpinellifolium. So this is indicative of an introgression. And then when you go outside that little blip, you see it goes back to the original topology. So by using this method, we were able to find 4 regions in the Hines reference genome where introgressions have occurred. So one is on chromosome 4. We're not really sure what that is. It might just have been happenstance. We don't know of any known resistance genes that are in that region. In chromosome 9, we have verticillium genes. So we were able to find introgressions from pimpinellifolium that introduced those genes into Hines. And then we have the I gene for fusarium wilt resistance on chromosome 11. So we were able to delimit the introgressions for each of these genes in Hines, which now is really useful because Hines is our reference for the other things that we're going to do. So we want to be able to rule out these introgressions that are occurring in our reference genome so that we don't think that they're something of interest in the population that we're actually looking at. Okay, so now I'll talk a bit about mapping a single gene using a mapping population with a close relative to Hines. So this was some work done by Sarah Hind and Greg Martin's lab. And it was really fortuitous because we had just sequenced yellow pear, and Sarah shows up in my office and says, we just found this interesting phenotype with yellow pear, and I heard you have the sequence. So it got off to a really good start. And so the phenotype is that yellow pear actually has reduced resistance to bacterial infection, which you can see here on the leaf on the right. You can see much more symptoms of spec than on the leaves on the left. So this was great because we had the sequence, so we can go ahead and try to find some candidate genes for this phenotype. And just to remind you, again, this tree is really blurry, but the 2 arrows are pointing to Lycopersicum and pimpinellifolium. So these are 2 very closely related species. And these are the 2 species that were used in the cross. So LE1589 is pimpinellifolium, and that is the same accession that was used in the tomato genome project. So we had sequence for that. We had the yellow pear from our previous work. And so Sarah crossed these 2 to develop the mapping population. And then she took all of the she took a batch of susceptible plants, which we used for the next step of the analysis. This was an overview of the pipeline that we used to identify the gene. So the first thing was to take those susceptible plants. So Sarah put them into batches of 13 plants in 3 pools. So those were then sequenced on 1 lane of Illumina, and those were mapped onto the yellow pear genome. So from our previous work in the Galapagos project, we already had the yellow pear genome. So we mapped these reads from her samples onto yellow pear. And then we called SNPs. So whenever we call SNPs, the SNPs that we find should be coming from pimpinellifolium. So they should be pimpinellifolium. And then we plotted these, the frequency of these across the chromosome. And then once we had a region of interest, then we went in to see what actual genes were there. So this is from her paper, and that's what the plotting of the SNP frequency looked like from pimpinellifolium as we go across the chromosomes. And if you look at, try to use this thing again, there we go. Chromosome 4, which is right where my red dot was briefly, you'll see that there's a reduced frequency of pimpinellifolium SNP and the frequency of the SNPs. And it drops off towards the beginning of the chromosome. So that was our region of interest to try to find the gene that's responsible for this phenotype. So the really great thing about that was that, as I said, we had the yellow pear, we had the pimpinellifolium. We went into that region and we looked for any SNPs that were causing a change in the coding sequence. And so what we found is that there was one gene in yellow pair that had some deletions that were causing a frame shift in the coding sequence. And that was only happening in yellow pair. It wasn't happening in pimpinellifolium or Heinz or the other accessions that we looked at. So this is a pretty good indication that that was our gene. So we developed some markers, and Sarah went on to do some fine mapping to confirm. And it does turn out that that was the actual gene conferring the phenotype. And then she went on to do a number of very elegant experiments in the lab to elucidate what this gene is actually doing. And if you want to know more about that, I'd highly recommend reading her paper. All right, so the next thing, another project is moving on to QTLs. And here we're mapping to an intermediate relative, so not as close to cultivated tomatoes, pimpinellifolium. And this was some more work coming out of Greg's lab, so a former postdoc in his lab, Zilong Bao, noticed that in this particular accession of Habrochytes, we have resistance to Pseudomonas. So you can see in the leaf on the left, looks nice and clean, and then you can see the really susceptible one on the right. And then there was also an intermediate phenotype here, which you can see in the middle. So again, keep in mind, this is a little bit more distant species that we're using in this study. So the reason that's important to keep in mind is that some of these issues with detecting recombination and then also using Heinz as a reference genome gets a little bit more tricky. So this is from his paper, an overview of how we did the analysis. So he mapped PTOS, which is essentially cultivated tomato, to Habrochytes, developed an F1 and then an F2 population. The F2s were barcoded individually and then sequenced, and then we aligned these to Heinz. We called snips. One of the problems we ran into is that the read depth was a bit on the low side, so we had to create a tool that would actually create bins of genotypes as you go across the chromosome. So since the SNP density was so low, it was oftentimes difficult to accurately call heterozygous sites. So by taking an average across the bin, we were able to determine what the overall consensus genotype should be for a region. So that was how we handled the low coverage data in this case. So then we took those average genotype calls and went on to perform standard linkage analysis with our QTL. So in doing that, we found a region on chromosome 2 and a region on chromosome 8. The region on chromosome 8 ended up being basically the whole chromosome, and there was very little recombination there. So he decided to move forward with chromosome 2 since it was a little bit more approachable. So we went on, similar to Sarah's project, to design some markers. So we used the Habrochytes sequence that we had from the resequencing, because there isn't a Habrochytes genome like there is for Heinz. And so we developed some markers based on his sequence, and then he was able to fine map to about a 1 megabase region on chromosome 2. And there were a number of genes, candidate genes there, that he did some further work on to look at expression. It was a little bit tricky though. So this project was obviously more tricky than the FLF3 project for a couple of reasons. First of all, we're looking at a QTL, so we don't have this single strong effect locus. And also, as I mentioned, it's a more distant cross, so we had this problem with reduced recombination in some of the chromosomes. And then also, there isn't a Habrochytes reference genome. So whenever you're doing something like this, if you don't have a reference genome for both of the parents, you don't really know what is in that segment there. So it's possible that there could be genes that are in there that aren't in Heinz. All of these gene models we're predicting from the Heinz annotation. So we know that oftentimes resistance genes, they can be rapidly evolving and they can change by copy number. So what would be of interest is to have a Habrochytes genome to see if those same genes are actually found in Habrochytes in that region. So this was a good experience for us to give us sort of the rationale for having a reference genome for other projects. So, sort of alluding to another project here. So knowing now what we know, that having a reference genome for the more divergent tomato species is really helpful. We moved on to a project with Persicoides. So this is a very divergent species, so it's actually an outgroup to the tomato clade. And you can still cross it with cultivated tomato, but it's a bit more difficult. And it's interesting, so this was also another nice accident. So there was a postdoc, there still is a postdoc in Jim Giovannoni's lab that was doing some work on the Persicoides introgression lines out in a field that they're currently using. This was a couple years ago, and it was really good conditions for spec that year. So Ari, the postdoc, was interested in looking at some fruit quality related things in the introgression lines. But since the conditions for spec were really good, he was able to see that there were some plants that were actually resistant to the spec. So now it's super interesting because it has two really cool things going on now. So we wrote a proposal to the Triad Foundation that we have at BTI, and were awarded a bit of money to pursue this project. So you can see here in the introgression lines where they've found various loci that are involved either in the fruit quality, so carotenoid biosynthesis, or pathogen resistance. So with the Triad, what we were able to do is make the genome for Persicoides. So we were able to sequence it using our funds, which was really great because it's really helped a lot of our downstream work. So what we did was we sequenced the genome at about 80x coverage using PacBio sequence. And then we corrected those reads. So if you're not familiar with PacBio, it has a very, very high error rate. So the first thing you need to do with it is map all of the reads to the longest, roughly 20% of the reads, and then correct those reads. So that's what you use for assembly. We tried two different assembly tools. So Falcon is known as the diploid assembler, and then we also tried Canu, which is a modified version of Celera assembler that's used for PacBio. So we ran two different assemblies, actually many different assemblies using two different tools. And we found, based on N50 values, average length genome, the resulting assembly size, that the Canu assembly was the better one. So we moved forward with that. And we did polishing using both the PacBio reads, and then also we had Illumina reads. So we mapped all those onto our genome and tried to remove as much of the error as possible, because PacBio often will leave indels. So those obviously you don't want to have. And then we took that polished assembly, and we used that. We sent that off to Dovetail to do Hi-C and also their Chicago libraries. And that was magical because whenever we got that back, it was chromosome length assembly. So we basically have our assembly in 12 pseudomolecules. Once we got that back, then we just did a little bit more work to fill in gaps, because the Hi-C will create gaps in the genome, which are just ends. So you can take your PacBio reads and again map them, and sometimes you can span those gaps and fill in the missing sequence. So this is what our assemblies are looking like so far, compared to the published Pinelii and the published Persicum genomes. So you can see that the Hi-C did a great job, because we didn't have really any good genetic maps to use for Persicoides. So the Hi-C was able to pull in all of these scaffolds and get our genome assembly up to pseudomolecule level. The longest sequence in Persicoides is 133 megabases, so that's basically a whole chromosome, which is really great. Contig size is huge, which is again good because we want to predict gene models. So if you don't have really long stretches of contiguous sequence, you're not really going to be able to predict your gene models very accurately. And the total length is comparable to what we expect the genome size to be for Persicoides. So we're pretty happy with our results for this genome. these are this is sort of a standard analysis that's done with genome assemblies and also annotations to give you an idea of how complete your genome assembly is. So these are genes, BUSCO, or a set of genes that should be universally conserved, and they should be present in single copy in the plant lineage. So you look for these in your genome to see how many of them you can find, and you can see so LA2951, that's like a Persicoides, and you can see that we have very, very few missing. We actually have fewer missing than if you run the same analysis on tomato 2.5. So it's not really statistically significant, but it's pretty cool. And then these three other assemblies in blue are other PacBio assemblies we did with coffee. So I just have those there as a basis of comparison. And overall, things are looking really good. We do have a bit of a higher duplication level in like a Persicoides of the BUSCO set, but I find that if I remove, so whenever you do the high C, you get your pseudomolecules, and then you get some other sort of contigs that weren't pulled in. So if I remove those, that number drops. And I think a lot of that stuff that didn't get pulled in is actually another haplotype that was assembled. Of course, there's also probably chloroplast in there as well. But that would be what would be causing this duplication, because like a Persicoides is very heterozygous, so we would expect that there would be some cases where the two haplotypes didn't get merged together in the assembly. All right, so of course, what people really care about with genome assemblies is finding the genes. So our next step was to run software called Maker. So Maker works by first you repeat mask the genome, because it's if you leave the repeats in there, it confuses the gene prediction software. So you repeat mask. And then you train some ab initio gene predictors. So we used Augustus and Snap. So you train those with a training set so that you have a model for what the genes should look like for the particular species that you're annotating. And you put all this into Maker. You also give it ESTs, RNA-seq data, all kinds of expression data, basically. You feed that into Maker, and then it'll give you a bunch of predicted gene models that are never 100% correct, but they're a good starting point of where coding regions should be. So when we did that, we got about almost 38,000 gene models. So this is comparable to what you see in Pinelli and like a Persicom. They should be pretty similar. And then we took these and then we tried to do functional prediction with those. So, what are they likely to encode? And we use this using BLAST sequence similarity, and then also interpro scan, which looks for domains in the protein. And this is a nifty little tool that we're really trying to encourage people to use. So as I said, with the annotation, you never have things 100% accurate. So one way of helping with that is to set up these instances of Web Apollo. And so users can come in and actually edit their gene of interest. So that's what this is here. So basically what you have are your gene model up at the top. That's the predicted gene model from Maker. And then you have tracks below that show all of your RNA-seq data. We had iso-seq here. Iso-seq is sequence that comes from PacBio sequence. And what's nice about it is it should, in most cases, give you a full-length transcript. So those are really good for annotation. We have RNA-seq from different tissues. And all you need is an account. So we have this set up for cultivated tomato too. If any of you are working on that and are interested in particular genes, you can contact us and we can set you up with an account. You can go in and actually change gene models and correct them. And all this goes into a database, and then whenever we release the next version of the annotation, all of your changes will be incorporated. And I just thought I'd throw this out there in case any of you are interested in Lycopersicoides. We will be making this publicly available in the next week or two. So this will be pre-publication. It'll be free for anyone to use as long as you agree to our access agreement. So, and it'll be on soulgenomics.net, Lucas Mueller's database. So you can let me know if you're interested in that. All right, so now that we have this genome, we want to move forward with the projects to look at these genes that are involved in disease resistance and fruit quality. So how do we do that? So we developed a pipeline for predicting, for creating markers to map traits in these crosses. So since Lycopersicoides and Lycopersicum are so divergent, oftentimes it's difficult to get enough recombination if you generate enough to actually map genes very well. So one of the things that was done was to cross Lycopersicoides to Penelii, which is a little bit closer. So we can actually get more recombination and fine map genes. So because of this, we made the pipeline so that it would give us markers that are for use in crosses between Lycopersicoides and Penelii and crosses between Lycopersicoides and Heinz. So we mapped Lycopersicoides to the published Penelii genome and to the Heinz genome. And then we used Mummer for that. And then we took those results and we identified any region where we had an indel that was greater than 30 base pairs, but less than 200. And those parameters can be tweaked, but that's what we used. And then we used primer 3 just to design primers on either side of that indel that we could use to distinguish differences in the F2 population to use as markers. And to confirm that these would actually work in PCR, then we took them and we blasted them back to the respective genomes. Because since the genomes are quite different, sometimes the primers wouldn't match 100%, or sometimes they would map too far apart or do something unexpected. So it's important to do that last step. And so in doing that, for Lycopersicoides and Penelii, we have about 1,000 markers. And for Lycopersicoides and Lycopersicum, we got about 600 markers. And the reason there's less for Lycopersicoides and Lycopersicum is because, again, they're more divergent, so less things aligned. And then also when we would blast the primers back, since we were predicting them on one genome, oftentimes those primers wouldn't match 100% in the other genome. So there's some tweaks we can make to the pipeline to pull out a lot more markers if we needed to. All right, so the last mapping approach, this is a work in progress as well. And again, a collaboration with Greg Martin's lab, is to see how we can use GWAS in tomato to look at novel disease phenotypes. So for this, there is a lot of data that's publicly available for various tomato lines and also wild species and pimpinellifolium. So we've taken advantage of this. So there's a paper where they sequenced 360 lines. Most of those were Lycopersicum. There's another data set of about 100 lines. Some of those include wild species. And then we also have SNP data and some other things that were just put into the SRA at NCBI. So we took all of this data and we generated this big tree from all of it. And this isn't that tree, but it kind of gives you the idea of this is a subset of those accessions, which is a little bit easier for presentation purposes. But it gives you some idea of how things sort of fall out. So it's how you would expect Lycopersicums cluster together. Then you have your land races down here by the star and heirloom lines. The blue is Cerasiforme. So Cerasiforme is generally believed to be admixture between pimpinellifolium and Lycopersicum. Then the green at the top, we have pimpinellifolium, and then the yellow is some more divergent wild species. So this was useful because now we can select from these clusters representatives to use for screening. So we want to get a broad picture of the diversity that's present in tomato based on this work with the resequencing data. So Sarah Hind, who was a postdoc in Greg's lab, and Samantha did work to select samples from this tree and then to procure seed for them. So I think right now in Sammy's office, they're running like a whole germplasm storage center. There's a lot of seed from this. And then Robin and Holly are also in Greg's lab, are involved in screening. So they're screening for novel phenotypes, pathogen phenotypes of Pseudomonas, responses to Pseudomonas. And what's really nice is that since we have all the sequence data, and now we're getting these phenotypes to tie in with it, we can try to use GWAS to get at the loci that are causing these interesting responses. So this is some work done by Adrian Powell. And he's been using, so the first thing that he tried to do was, we know FLS3 is involved in pathogen response. We have a sequence from a number of individuals who are non-responders, a lot more who are responders. So taking all this sequence data and the phenotypes that we already have, he did some work to see if we could actually detect a known gene. And you can tell from the Manhattan plot, he was able to do this. There is a bit of noise, but you see this nice strong peak on chromosome 4. And if you zoom in to where that is occurring, that actually is at the FLS3 locus. So right now, Adrian's working on fine-tuning this, and then he'll be interested in applying this to the new phenotypes that are coming out of the screen. And the last thing I wanted to talk about briefly is a little bit of advertising for one of our group members. So he wrote this program called SnipBinner, and this is useful for the creation of bin maps. So if you have GBS data or RNA-seq data, and you generate a lot of SNPs, oftentimes that can be a bit too much information. So bin maps are a way of sort of reducing the complexity of that data. So the first thing it does is so in this case, it's been tested with RILs. So it takes each line and it looks for the crossover points in each line. And then based on an algorithm, it takes those crossover points and constructs a bin map from that data. And also the other thing about it, there are some other tools that do some similar things, but this one, if you have a BCF file, it basically will take BCF files from any SNP color. And this is just a little overview of basically how it works. So at the top, we have the RIL. So first, it does a step that I'm not showing here, where it uses some hidden Markov models to identify the genotype. So genotype calling can be messy, so it does some work to sort of smooth those out and determine the recombination breakpoints, which you can see at the top. And then it uses an algorithm to take those recombination maps and collapse them into a bin map, which can then be used in QTL mapping. So this has actually been used in a paper that we hope will be coming out soon by Itai Gonda and Jim Giovannoni's lab. So he used SnipBinner to, in his analysis of some fruit quality traits in a Pimpinellifolium tomato RIL population. So that will be coming out soon. In the meantime, if you really want to play with the tool, it is publicly available on GitHub. So you can go there and try it out. So far, we've only used it on RILs, but I believe that Itai has been testing it out on an F2 population, actually the F2 population from the Habrokaites project that I talked about. So if you're interested in using it on F2, we can touch base with him and see how it's working for that. Okay, so nearing the end here. So as I said, I work as a bioinformatician, and I'm very, very just absolutely thrilled that I get to work with all these wonderful people in molecular labs that are willing to share their data with us and let us mine their data for exciting things. So as you probably noticed, a lot of this work was done in conjunction with Greg Martin's lab. So specifically, I'd like to thank Zilong Bao, who was the lead on the Habrokaites project, Sarah Hind, who was the lead on the FLS3 project, Samantha, who's doing the work with the mapping of the Persicoides genes, and then also Robin and Holly, who are doing this big screen and working with Adrian on the GWAS. In the Giovannoni lab specifically, I'd like to thank Ari, who's done a lot of the RNA sequencing for like a Persicoides genome, which allowed us to do the gene model prediction. Itai, who worked with David on the SNP project, and Yumin, who is awesome at RNA extraction and gave us really high quality RNA sequence or RNA for our isoseq sequencing for the annotation. Also the Mueller lab, who is always extremely helpful, and obviously I spent a lot of time there, and the Galapagos work was done while I was a postdoc in his lab. Our collaborator, Bjorn Usedel, and specifically Alessandra in his lab, who has helped with the Persicoides genome project. Roger at TGRC, who always is sending us seed and things that we need to get our work done. And of course, the BCBC group, which, as Joyce mentioned, is newly formed. So David, who wrote the SNP software, and Adrian, who is doing the GWAS analysis, amongst many, many other things. And so here's David's in the left corner there, and Adrian's on the right, and then there's some weirdo photo bombing in the middle there. And this is a really exciting day for us because this is when we got our first BCBC server, and that empty slot that we're peering into is where our new 1 terabyte memory server is currently residing. And it's working very, very hard on assembling a milkweed genome for George Jander right now. So it's an exciting day. And then of course money. It's good that people give us money to do things. So huge thanks to BTI, who's supported a number of the things that the BCBC group is doing. The NSF that occasionally does hand out money. And the Triad Foundation for the Persicoides project. And one last thing, one last advertisement. For any of the grad students and postdocs, or really anyone, if you are interested or want to know more about bioinformatics or have a problem with your project, we run this bioinformatics hour at BTI on Wednesdays 1 to 2 and Thursdays 10 to 11 in the resource center where the fish tank is, but I hear it's going away. So if you want to stop by during those times, it's free. You can come and talk to us, and maybe we can help you. And also, we'll be starting the BTI Bioinformatics course on March 13th. So, and these are the topics that we'll be covering. So if those are things that are useful to you, that you'd like to know more about, just send me an email and I can let you know the schedule and sign you up. So I think that's it. Thank you for your attention. Yeah.
About the GWAS project. So GWAS typically is a diversity panel within a species, whereas you're sort of proposing a very diverse panel that crosses species.
So for the GWAS that I mentioned, that was just looking within a Persicum. Yeah.
So you're not saying you're going to try to do GWAS within that whole.
I think Adrian, who is sitting behind you, and he can correct me if I'm wrong, but I think we're trying to stick to a Persicum because whenever you add pimpinellifolium, then there's structure issues and all these other things. So, yeah. Other questions?
This has been a production of Cornell University, on the web at cornell.edu.